Hello everyone. Welcome back to Dr. K. Primer video lecture series presenting by Dr. K. Prem. That's me. In today's lecture, we'll talk about cohesive ends and blunt ends. The only difference between the cohesive ends and blunt ends are the shape of the complementary strand. The shape of the complementary strand. In case of cohesive ends, one of the strand is little longer than its complementary strand. You see, 5 prime to 3 prime, 3 prime to 5 prime. The 3 prime to 5 prime strand is 4 nucleotides longer than the 5 prime to 3 prime strand. So these 4 nucleotides are unpaired. These 4 nucleotides are unpaired, right? So the end which is having the unpaired single strand nucleotides are called as cohesive ends. And the two strands are unequal in length. One of the strand is little longer than the other strand. When it comes to the blunt ends, both strands are in equal size, right? You see here, end is having two strands are in equal length. There is no single strand overhang unpaired nucleotides, right? Hence, they are called as blunt ends. So the blunt ends are equal in length, whereas cohesive ends are not equal in length. They have unpaired nucleotides, they're single strand, right? So we will see the definition of cohesive ends and blunt ends in the next slide. Here this is a cohesive end and this is a blunt end. Here we can see the definition of cohesive end and blunt end. Cohesive ends. To release the DNA fragment from its source, two phosphodiester bonds breaks are required. Two phosphodiester bonds breaks are required, right? So one cohesive ends cut at one end between two bases on the same strand. The second cut at opposite end of the complementary strand. One cut at one end between the two bases on the same strand, and second cut at opposite end of the complementary strand. A 4 nucleotide overhang at one end of the DNA fragment form, which is ready to form a hydrogen bonds and anion. These are called sticky ends. Blunt ends cut between the two bases at same place in the duplex DNA leads to symmetrical breakdown of the DNA. So in cohesive ends, two phosphodiester bonds are breaking at different places. They're opposite ends. That's why they are forming the single strand overhangs. But blunt ends are two phosphodiester bonds are breaking at same place in the duplex DNA, leads to the formation of symmetrical breakdown of the DNA. We'll see how they are forming in the next slide. This is the linear DNA, which is having two restriction sites. One is EcoR1, this is a restriction site, and this is a small one restriction site. So here I'm showing the phosphodiester bond. This is one of the phosphodiester bond. You all know that in each strand of the DNA, nucleotides are connected by the phosphodiester bond. Unless you break down the phosphodiester bond from each strand, then only that DNA fragment is released from that source. Otherwise, no. Right? So phosphodiester bond is the backbone of DNA. You see. So phosphodiester bond is indicated here, magenta color, right? This is one of the phosphodiester bond. Ecoorganism in the digestion, restriction endonucleases are first they'll scan the DNA and binds to its restriction site, then followed by the cleavage. So scanning, binding to the restriction site, then followed by the cleavage. We'll see them. Ecoorganism identified its restriction site. That's a GAA ETC. Then SMA one identifies its restriction site. That's a CCC GGG. Then ecoorganism digests the two strands and leaves the cohesive ends. You can see here first cut at the one end. This is one end between the two bases. Between the two bases, you see, first cut at the one end between the two bases of same strand. Two bases of, this is the one strand. Here you can see first cut between the two bases of same strand. First cut, right? This is the first cut. And the second cut, opposite end. This is the one first cut. Second cut is at the opposite end of complementary strand. Opposite end of complementary strand. You see? Because of that, you get a cohesive ends. Means two strands in equal length. One strand is having 4 nucleotide extra. That's a single strand and unpaired. This is called as a cohesive end. And you see here SMA one digest the DNA two phosphodiester bond at same place. One first phosphodiester bond breakdown and second cut, right? Then releases the strands. Here you can see these are the because of SMA1 digestion, and the two strands are in equal length. So at termini, they don't have an extra unpaired single strand nucleotides. Hence, they are called as blunt ends, right? Here, because of the ecoorganism digestion, you can see cohesive ends. Because of SMA1 digestion, you can see blunt ends, right? Hope you understand this lecture, right? Thank you for listening. If at all you like it, just try to subscribe my channel. That's Dr. K. Prem Primer. See you all again. Until then, bye-bye.